Good evening, everybody. Today we shall start Block 3, Conservation of Biodiversity, and it has 4 units, Unit 9, Unit 10, Unit 11, and Unit 12. Unit 1 is about the introduction, Unit 10, Conservation through protected areas, and Unit 11, different methods of conservation like in situ and ex situ conservation, and Unit 12, social approaches to conservation. Conservation of biodiversity. So, conservation biology, it is a branch of biology, or it is a field, and it gives an idea about how to manage and conserve biodiversity. So, the goal of conservation of biology is to establish effective ways to conserve species and their habitats. Wrongly, it is again printed as species and their species, but it will be habitat. So the goal of or the aim of conservation of biology is conservation of species and their also their habitat. So this is a brief description about different units. So first we shall go to Unit 9, Introduction to Conservation Biology. So conservation biology, it is a multidisciplinary science, and it was developed to deal with the loss of biodiversity, because recently there is loss of biodiversity. So that is why to stop the loss of biodiversity, the conservation biology so conservation biodiversity, this is the term refers to all biological variations that exist at all level. We have already studied before about what is biodiversity. Biodiversity in different levels, genetic level, species level, and ecosystem levels. So conservation biology, it has two aims or objectives: exploring human impact on biodiversity. So because there is the loss of biodiversity due to human beings, so there is great impact of human beings or humans on the loss of biodiversity. So how human activities they impact biodiversity. Then next is to develop practical ways to prevent the extinction of species. So when there is extinction of species, there will be loss of biodiversity, and mostly it is due to the human factor, due to human beings. So we shall, it has two aims. What are the impacts, to study about the impacts of human beings on the loss of biodiversity, and next is to develop practical ways to prevent the extinction of species. So this is the history of conservation biology. Conservation biology was introduced by Dasman in 1968, and Ehrenfeld in 1970. Conservation biology emerged in the 1970s and 80s from the old biological science, more established in response to the growing rate of extinction and the diversity of life on Earth. So, biological science, it is an old science, and when it was noticed during 1970s that so there was extinction of species and loss of biodiversity of life. So the a new branch of science, it was established, and so we call it the conservation biology. So the ultimate goal of the conservation biology is to establish effective ways to conserve species, and there it is written again here, it is species, but so it is actually the habitat. So the ultimate goal is to establish effective ways to conserve species and their habitats. So in 1980, World Conservation Strategy, so that is the first international document for diversity conservation. So internationally, say document, it was prepared for biodiversity conservation, how biodiversity will be conserved. So there is a document prepared internationally, and that is known as World Conservation Strategy. So this it has so report, it was prepared taking into account various reports and studies on biodiversity loss and biodiversity conservation, and they prepared a report, and this report contributed to our common future. So what is common future? So all the organisms, we have a common future. So if other organisms, they will also die, then there is also no future for human beings. So that is known as common future. That means it is common future for all the organisms on the earth. And this report, this is known as Brundtland report, was prepared in 1987, and this laid the foundation for the goal of sustainable development. So today we know about the sustainable development, and this the goals. What are the goals? Different goals of the sustainable development, and these goals of sustainable development. So uh, so it is its foundation in the Brundtland report, and so it is very important. So it was prepared by different institutions like International Union for Conservation of Nature and Natural Resources, UCN, Food and Agriculture Organization, World Wildlife Fund, UNESCO, so like this. So the uh, objective of the uh, World Conservation Strategy, or WCS. So there are three main objectives of WCS. So WCS, or World Conservation Strategy. So first one is maintaining vital ecological process. So, so for living organisms, or for conservation of biodiversity, the ecosystem that is very, very important. And in ecosystem, there are different organisms, and there are also non-living materials, abiotic and biotic components. And when the abiotic components and biotic components are present, and there is nutrient cycling and other activities of the ecosystem, then also the living organisms, they can survive. So first is maintenance of the ecological process. Then the maintaining the genetic diversity. So previously we have also discussed about the genetic biodiversity, what is the importance of genetic diversity. So one of the objective is how to maintain the genetic diversity of plants, animals, and every living organisms, like the crops which we cultivate, and the and which is useful for science and medicine and technology. Then number 3 is ensuring sustainable use of biodiversity and ecosystem. So, for maintaining biodiversity, sustainable use of the biodiversity, that is very, very important. So because the human population, it is growing, and human population, they also need many materials, the resources, natural resources, and the natural resources, biotic and abiotic resources, they are required by human beings for different purposes, industries, food. So we have to use biodiversity and ecosystem, but there must be sustainable use so that they can also be preserved for the future generation. So if we use without considering the future generation, then the biodiversity and ecosystem, it will be lost. So we have to use biodiversity and ecosystem, but there must be sustainable use. Then the multi-dimensional aspect of conservation biology. So conservation biology, so it is a multidisciplinary subject, and encompassing different branches like taxonomy. Then you have to study the functional aspects of the ecosystem, how the ecosystem functions. Then the phylogenetic, how the phylogenetic means the evolutionary history of the living organisms. So not only we have to identify the organisms, which organisms are present in the ecosystem, but also we have to know the evolutionary history of the living organism. Then the genetic, that means we have to know the genetic relationship between the living organisms, how one organism is different genetically from another organism, or how they are related genetically. So these also we have to study. Then we have to study also the landscape, because the landscape, they contain the living organisms, biodiversity, and many other elements of variability of life on Earth. So this is, we have to study the variability of life. So we have to study many of the aspects. So the variety of organisms which we find or the biodiversity. So it is produced through evolutionary process, and it leads to formation and cohesion of organisms into species. And through the evolutionary process, the different species they are produced, and we can recognize these species. So species, so that is the basic unit of classification, and that is also unit in conservation biology. So in conservation biology, we study about the species richness. Previously, we have also discussed about species richness. The species richness with regard to a given community. So, in a specific habitat or in a specific ecosystem, so you'll find the non-living submaterials or the abiotic component, and the biotic components are all the living organisms. So all the organisms which are present in a specific habitat or specific ecosystem, so they are known as a community. That means a community, it has different species, many different types of species. So the species, so there species, there may be plants, the species, there may be animals, the species, there may be the microorganisms, all the organisms which are present in that community. And these communities, they interact. So a group of interacting species in a given place. And these species, they interact. One species interacts with another species. So this interaction, it may be in terms of food or shelter or predation. One organism, it will eat another organisms, or any type of relationship, the relationship between two different organisms. So in a community, the organisms, many organisms are present. Not only they are present, but they also interact. So interact species, and that is known as the species richness. That means how many number of species present in a given place is the species richness. So we shall also study the species evenness, not only species richness, but also species richness. That means the how much this species evenness or species abundance. So that is depends on the number of members of a species. So, so there is an example. Suppose we have two species in which first has three members. So, but this second has 700. The second community of species is far more even than the first. So, when there are more numbers, then the species evenness that is more. So the diversity can be studied as a number of species present, how many number of species are present, and the number of interaction, not only the species present, the number of interactions between the species, what type of organisms are present, what are their role in the ecosystem. So these interactions and the stability of the ecosystem So this can be understood in a variety of ways from notions of stability written from time after a perturbation. So stability. So when there is species richness and species evenness, then the ecosystem it will be stable. If the ecosystem it is not stable, the organisms they may not survive. So this how can we understand stability? So this is by the return time after a perturbation. Perturbation means if there is any disturbance in the ecosystem. For example, if there is a wildfire in a forest, so there is a fire. So this is an disturbance, and many organisms they will die. But after it will take a time to return to its uh, original. So this return time that is known as stable. If it return to normality in a short time, then it is very stable. And if it takes a very long time, the stability it is less. So previously we have discussed not only we have to study the species. If we want to know the species diversity or species richness, species evenness, then we have to identify the species. So how can you identify? We identify by taxonomic methods. So these taxonomic methods, we identify the species, and not only we have to study the species, what are the types of species, what are the interaction of the species, but also we have to study their evolutionary history, and this is known as phylogenetic information, phylogeny. So we have to consider and we have to include the phylogenetic information when we study when we measure the biodiversity. So how whether the biodiversity is less, the biodiversity is more. So we have to study the number of species, that means the species richness, then the how many members are present, that is species evenness, and also we have to study the evolutionary relationship between these organisms and know the evolutionary history of that particular species. So for studying the phylogenetic information, so we study the phylogenetic diversity. So when there are a number of species, then we have to study their relations, evolutionary relation. So all the organisms they are related through evolution, but some organisms they are related very closely, and some other organisms they are related distantly. So if evolutionary through evolution, if they are related closely, then so the two species they will also be very close morphologically and taxonomically. So if we find there is a taxonomic similarity between any two species, then we understand that they are very close, by evolution also. And when they are very differently, they are morphologically they are different, taxonomically they are not very close, they are different, then they are also different through evolution. Evolutionarily also they are very different. So if we evolutionary studies or phylogenetic studies, we find that there are many differences, then that is known as phylogenetic diversity. Just like we study the species diversity, that means what are the different types of species present, that is species diversity, and the interrelationship between these species, how they are related, if they are related closely or if they are related distantly, and this is known as the phylogenetic diversity. So in phylogenetic diversity study or PD, so the phylogenetic diversity study, so we have to study the traits or the characters present in the species. So how we identify or study a species? From its traits or the characters. So if the organisms they are very closely related, then they will share some of the traits. They will share some of the traits. Then if there is a group of organisms in a and this group of organisms they share some of the traits, then this group of organisms because this group in this group all the organisms they share a share some traits, then we we call this group a clade, CLADE, clade. A clade, that means it is a group of organisms and they share some traits. They share share some traits, then it form a clade. Then we have to identify different clades. A clade, in a clade, the organisms they are closely related because they share. So all the organisms in that group they share some traits. So that is why our character they share some characters. That means they have some common characters. All the organisms of that group, so they will form a clade. So in a ecosystem, we can find many such clades, different clades, and so there is also some relationship between different clades. First, we have to identify the clades. Clade. A clade, it is a group of organisms. They share some of the characters which are common and which are unique characters. And these characters, they are not shared by other organisms. So they form a clade. So we'll find different clades. So we have to find out the relationship between different clades. So for studying the relationship between different clades, we have to prepare a cladogram. And there is another one is phylogenetic tree. So it is a phylogenetic tree. It is a graphical representation. It looks like a tree having branches, and so and it gives an idea about the evolution of different species, how the species that they have evolved, the evolutionary history, and also it gives the relationship. So in that phylogenetic tree, if two organisms they are closely placed, so the phylogenetic tree it has different branches. So if two organisms they are placed closely in that tree, then these two organisms they are closely related through evolution. If they are placed distantly, then the relationship it is distant. So uh, so the evolution through evolution, um, one species produces one or more species through evolution. So there is a ancestor and descendant relationship. So if there is a species A and it produces through evolution, two new species are produced. So they are species B and species C. Then the species A is the ancestor, and species B and species C they are the descendants. So this uh, ancestor descendant relationship, so you can study from a phylogenetic tree. So the cladogram and phylogenetic tree, so they are very similar, and because but so there is a difference. The phylogenetic tree it also includes the ancestor descendant relationship, and apart from merely relations of similarity. So in cladogram, so the different organisms they are organized, they are represented in a graph according to their relationship. Relationship means if there is there is a relationship in taxonomy, or there is similarity in DNA. So then this is known as similarity. So the cladogram it is prepared. So according to the similarities and differences in the characters. But the phylogenetic tree, so this is also based on the similarities and differences. But from the phylogenetic trees, we can also study the phylogenetic relationship. That means the relationship between the ancestor and the descendant. So the phylogenetic diversity, from the phylogenetic tree, we can study the phylogenetic diversity. So in the diagram, you can see this is the phylogenetic tree, and from this we can calculate the phylogenetic diversity, so which is equal to the sum of the length of all those branches that are members of the corresponding minimum spanning path. You can see in the diagram. So this is 4 species, or this actually in phylogenetic tree we study taxa. That means it may be a species or it may be a genus or it may be family. So these are different taxa. Species, that is also a taxon. Species is a taxon, genus is a taxon, and family is a taxon. So we not only study about the species taxon, but we can also study genus and other taxa. So here in the diagram, you can see 4 number of taxa, and this is 2, this is 6, this is 8 and 10. So if we have collected many, there are many species, and we have to study, you want to study the relationship between these 4 species or 4 taxa, 2, 6, 8 and 10. Then, so in this tree, phylogenetic tree, so there is a branch point, and the point from which a new branch arises, that is known as the branch point. So it is a branch point. So branch point indicates that the new species has evolved. So there is a straight line, and this straight line, it produces a branch point. When there is a branch point, that means there is evolution and it has produced new descendants. So if at the base you can find the descendant, that is descendant number 10, and this is the branch point. So if it is, so if when we study the relationship, so between the descendant, the number 10, which is written in the graph, it is a descendant, in the branch point is the ancestor. So at the branch point is the ancestor, and at the tip of the branch, so this is the ancestor. So you can find the branches having different length. So you can see here the species 10 or taxa 10. Then next there is a branch, it is small. Then next the branch number 8, it is longer than the 10. Then there is a branch number 6. Then another branch, another small branch. And so this is 2. And if the branch it is small, that means so there is a very similarity. If the similarity it is less or differences are more, the length of the branches they will be more. So the length of the branches, if the length of the branches it is more, then there is more difference. More difference mean evolutionary they are very different. So that means there is more diversity. So diversity, that is also more. So in branch number 8, the diversity it is more, and in branch number 10, the diversity is less. So if we have to calculate the phylogenetic diversity, and we have to calculate all these diversities. So ten. So evaluation of priority for conservation of habitat and species. So first we have to evaluate what is the priority for conservation. So there are different methods of conservation. We have to study the priority which habitat or which ecosystem we have to conserve for conservation of habitat and species, and we also have to identify the species and what are the habitats. So we have already discussed the human activities, that is the important factor, the largest factor which is which causes biodiversity loss. So in response to growing concern about future diversity, 10th Convention on Biological Diversity developed a strategic plan for 2011 to 2020. So there are from time to time there are different conventions on biological diversity. So on the 10th convention, 10th number of convention, so that developed a plan, and this plan it was for 10 years from 2011 to 2020. So they introduced protection targets. So they have given targets. So they have given a target, and you have to achieve that target. And this target, that is also known as Aichi target. So this Aichi is a district, we can call it a district in Japan, and this convention it was held in that district, Aichi district. That is why that is known as Aichi target. So this uh, Aichi target, it gives a target for conservation. So one primary target is to protect 17% of the global land surface. So what is the target? So we have to achieve that target in 10 years, that is from 11 to 2011 to 2020. It was given a target to protect 17% of the global land. So you have to protect 17% of the land or habitat, and focusing on areas of importance for degradation and biodiversity and ecosystem services. So for protection of biodiversity, and it also focuses on ecosystem services. We have discussed earlier in block 1 about the what are the ecosystem services, and it was also repeated in block 2. So, all the habitats, they have different values according to biodiversity. That means biologically, some habitats they are very biologically very important, and other habitats they are biologically uh, uh, less important. So the if they have high importance, then they have a high value, and these are known as high value habitats. So first we have to find out the what are the we have to locate what are the high value biodiversity habitats. So according to the estimates, so only 18.6 of high value habitat is currently protected globally at present. So only 18.6% of high value habitat it is protected. That means more than 80% of habitat it is not protected. So if it remains unprotected, there is a chance of the loss of biodiversity. So in developed countries, the red list of endangered plants and animals have become an important standard tool for conservation science, policy, and action. So we shall study afterwards the red list of endangered uh, organisms prepared by IUCN. IUCN has prepared and prepares the red list of endangered organisms. And in developed countries, they focus mainly on these endangered plants and animals which are present in the red list. So um, these species they can survive only in their native habitat. So if we see some species they are present in a particular habitat, then they are adjusted and adapted to that particular habitat, and they can survive only in that habitat. And if the habitat it is lost, then the organisms they will also not survive. So uh, when there is conservation, not only we have to focus on the endangered plants and animals which are present in the red list, but also we have to focus on the habitats in which they live. So if in a habitat many organisms are present, there are some organisms which are very sensitive. That means if there is some change in the habitat, they will not survive. And these species, they are sensitive species. And if in that habitat we find a sensitive species, If we find sensitive species, that means we know that the habitat it is healthy. So presence of the sensitive species that indicates that the habitat it is very healthy. Next is selection criteria for protection of species. So IUCN it recognizes many different criteria for protection of the species. So what are so there are many mechanisms for selection. So you have to select, then categorize, and zoning of the protected areas in the world. So uh, for protection of the biodiversity, we have to protect the habitats in which they live. So if we protect some area, the natural area in which the organisms live, and that is known as a protected area. So the protected areas, they are legally established sites. So, these are according to law. The laws passed by parliament or any body that this is that this is a particular this area and it will be protected. So protected areas they are legally established. That means they are established by law, and these sites they are managed for conservation. So throughout the world, so there are more than 8,000 protected areas. So for protection areas, we have to first we have to select. So there are different criteria how to select a particular area as a protected area. So throughout the world, there are 8,000 protected areas and which cover 750 million hectares of marine and terrestrial ecosystem, not only on the land but also in the ocean. So, this accounts to 1.5% of Earth's surface. That means it includes both terrestrial and marine. If we include all terrestrial and marine, that it accounts 1.5%. And if we consider only the land, then it is 5.1%. So there are different types of protected areas, and we shall study later on afterwards. So, but now they comes to the methods to select a protected area. How to select a protected area? So the gap analysis uses geographic information system. All of you must be knowing what is a geographical information system, and it gives about the geography of a particular area. And we have to, what is gap analysis? Gap analysis identifies the gaps in biodiversity protection. So if there are, in a country there are 10 protected areas, that means it do not cover all the area. So if there are only 10 areas and there may be some gaps, that means there may be some areas and which are not protected, and we have to identify those gaps, and that is known as gap analysis. Okay. Now we come to IUCN guidelines for red list categories and criteria, red list of Indian flora and fauna. So IUCN, International Union for Conservation Nature and Natural Resources, IUCN, it has prepared a list of organisms, and that list it is known as a red list. So this indicates how close a species is to becoming extinct. So in the diagram you can see at the top it is written extinct or EX. That means they are not found at all. So these organisms once they were present on Earth, but they are now not present. Like the, for example, common example is and dinosaur. So dinosaurs they are extinct because once they were living, but they are not present today. So these are extinct. So a red list. So this indicate how close a species is to becoming extinct. So extinct, below the extinct you can find extinct in the wild. Then below that critical endangered, below that endangered, below that vulnerable, below that near threatened, below that least concerns, like this. So this indicates how close they are to becoming the extinct. So in the extinct it is given a symbol plus, and it is when it is distant, more it is more distant, then it is at the other end it is negative. In the negative it is written green, it is green colored, and you can find it is written least concern. That means today there is no chance, there is there is no chance of becoming extinct. If a category it is near the extinct, that means it is nearest is extinct in the wild. It is very close to become extinct. Then next comes the critically endangered. Then next comes endangered. Next comes vulnerable. Next comes near threatened. So like this. When a category it is very near, then it is very near to extinct. So species are assessed against 5 criteria. So there are 5 criteria. It is there: the rate of population decline, the geographic range, where the species already possesses a small population size, where the species is very small or lives in a restricted area, whether the result of a quantitative analysis indicates high probability of extinction in the wild. So these are the 5 criteria. So these 5 criteria so these criteria, they will determine the category. So if we have to study these criteria, then the IUCN it will place in one of the categories. So it may place in near threatened, or it may place in critically endangered, or 18th place, or it may be in least concern. So first we have to study these 5 criteria. So species in the vulnerable, endangered and critically endangered. You can see here vulnerable, which is yellow in color, then endangered, saffron or orange color, then red color, that is critically endangered. These 3 categories, they are included in threatened category. So 3 categories come under threatened category. Threatened category means it has 3 subcategories: critical endangered and vulnerable. So the IUCN red list doesn't include not evaluated species. So actually, which is the category which is present below, not evaluated, that means the IUCN it is not evaluated. So if IUCN it is not evaluated, then it will not include. Actually, it is given in the list, but according to IUCN list, so there is no not evaluated list. Because it is not evaluated, so there is no not evaluated category. That the critically endangered species may be regarded as possibly extinct or possibly extinct in the wild. So critically endangered. So if there is a possibility that if we do not, we were not confirmed that it is extinct, but there is a possibility that it is extinct, then also it is kept in the group critically endangered. So for regional, so IUCN it is no, there is no category of not evaluated. For regional assessment, 2 additional categories are also available. For if we want to study in a specific region, that means if in a specific country, or if you study in a specific state, then that is known as regional. So if we study, if we assess in any region, small region, then you can include not applicable and regionally extinct. Regionally extinct means, that is extinct means regionally extinct means it is extinct only in that region, and they may be present in other parts of the world. Just like the cheetah, which were extinct in India, in Asia, but they are present in Africa. So they are regionally extinct. That means they are extinct only in India. If we study a particular our country India, then we will keep cheetah in regionally extinct. Actually, it is not extinct because they are present in Africa. But in India, if we study only the region of India, then the regional, if we assess, there is a regional assessment of India, then we can give one category, that is another extra category, that is regionally extinct. That means extinct in India. So these are the 5 criteria uh, for categorization of different categories. So these species, it has to be evaluated thoroughly. And according to these 5 criteria, these species, it has to be evaluated and it has to be kept in one of the categories. So there may be 5 so these if you strictly follow the categories of IUCN, then 1, 2, 3, 4, 5, 6, 7. There are 7 categories, and you can, if we study the regionally, then can you add another one more category. So you can keep any category, but we have to study the species and evaluate as per these 5 criteria. So these are the criteria. Extinct, the applies to a species where the last individual has died or where systematic and time appropriate survey have been unable to log even a single individual. So if there were many members of this species, but the species they all died, and the last member also died, and there is no any member found living, then we call it extinct. And after survey also we cannot find any single member. If no any single member is living, or after thorough survey or in investigation we cannot find any single member, then we call it extinct. The next is extinct in the wild. Contains those species which members survive only in captivity. If a few members are found in zoos or they are kept by human beings, but they are not found in the wild, then this is known as extinct in the wild. So there may be a few members living, but these members, if they are living only in the zoos, then it is known as extinct in the wild. Then critically endangered. Like critically endangered species, they have a very high risk of extinction. So because the population decreases, so if the population, rapid population declines of 80 to more than 90% over the previous 10 years. So if within the 10 years, when previous 10 years, means if we study today, it is 2025, then from 2015 to 2025, If within this 10 year of period, if 80 to more than 90% of the population or the members they decrease, then we keep it in critically endangered category. It may be 3 years, it may be previous 10 years, or if the lifespan of the organism it is very less. That means if the lifespan it is only 3 years or 5 years, then we have to consider the generation. So 3 generation, we have to consider 3 generation. If in 3 generations or in last 10 years, so if the population declines 80 to more than 90%, then we keep it in critically endangered category. And if current population size of fewer than 90 individuals, or if the total number of individuals in world it is less than 50, then also we keep in critically endangered. Then endangered. So if the population decreases 50 to more than 70% within 10 years. If it in 10 years if it declines to from 80% to more than 90%, then it is critically endangered. If it declines more than 50 to more than 70%, then it is endangered. Or if the population size it is 250, less than 250. If the size, the number of individuals throughout the world it is less than 50, then it is critically endangered. If the number it is slightly more than 20, more than 50, but it is less than 250, then it is endangered. Similarly, vulnerable. If the population declines 30 to more than 50%, then it is vulnerable. Or the current population size it is less than 1000. The number of individuals throughout the world, there may be less than 1000, but more than 250. If the number becomes less than 250, then it is endangered. But if it is more than 250 and less than 1000, then it is vulnerable. Similarly, near threatened. So near threatened means it has very high possibility to become extinct in near future. Very shortly, there is a possibility that it will become extinct. Then it is near threatened. Then another category, that is least concern. Category containing species that are pervasive and abundant. So if we assess any organism, any species, that it is found in very large number. So if it is found in very large number, then there is no problem. Least concern means there is no problem. Then there is another category, that is data deficient. So that means there is a study, there is a assessment. So to keep in any category, we have to assess according to these 5 categories. We have to evaluate thoroughly then only you can keep in any of the groups, 7 groups. But data sufficient means the study it is not sufficient. So the amount of data available is less. So then we don't know to which category we can keep. So it has to be kept in a category that is data deficient. Then not evaluated. So this not evaluated category that is not found in the IUCN red list category. So there is no such category like not evaluated. And not evaluated means so this category includes any of nearly 1.9 million species described by science. So science or scientists they have identified and described more than 1.9 million species, but all these 1.9 million species they are not assessed by the IUCN. So IUCN cannot keep it in any group. So these 1.9 million of species they may be evaluated by scientists in a different laboratories or different scientists they may have identified. But so the assessment as per the criteria of IUCN, the IUCN it has not studied all these 1.9 million species because it is difficult. So the species which it has not evaluated, it is included on under not evaluated category NE. So we can keep in any category to a plant if it is not categorized, but there is no such category according to IUCN red list. Then selection criteria for protection of habitats and hotspots. So for conservation of the biological hotspots, there are 4 criteria, and these uh, criteria this contains unique and rare habitats. The habitats it must be very sensitive, fragile. That means if there is slightly modification, so the ecosystem it will not survive. So these are the areas. So then what are the biodiversity hotspots? So currently there are 36 recognized biodiversity hotspots. These are Earth's most biologically rich yet threatened terrestrial regions. So just we take a break for 5 minutes, then we'll again start. Okay, welcome again. So conservation of biodiversity hotspots, they are very, very important. So what are the hotspots? So there are two criteria. So there are two important criteria. The hotspot, it must fulfill these two criteria. One is uh, it must contain at least 1500 species of vascular plants found nowhere else on Earth. So we have to study the only the vascular plants. That means in a hotspot, so there may be different types of biodiversity like plants, animals, microorganisms, and among the plants there may be um, bryophyta or algae or like this. But we have to study uh, the number of the vascular plants. Vascular plants, the plants which have conducting tissue or xylem or phloem. So these are the vascular tissue. These are known as the vascular plants. And so there is the hotspot. It may contain at least 1500 vascular species which are endemic. Endemic means so these vascular plants, they are present only in that specific area and they are not found in any other place of the world. So if they are not found in anywhere but they are present only in that area, then it is known as endemic. So it must contain at least 1500 species of vascular plants which are endemic. And it should have lost 70% of its primary native vegetation. Primary native vegetation means the vegetation who uh, mm, is present in naturally. So that is the natural vegetation of that area. If it has lost about 70% of its natural vegetation, That means it must qualify two criteria. One is it should have high biodiversity, and in the biodiversity, if an area it has high biodiversity, then it must contain endemic species. So it should have at least 1500 number of endemic species, and so there is loss of more than 70% of its native vegetation or the natural plants. So the hotspots, they are not formally recognized. So the protected areas, so there are different types of protected areas, and already I have told you these protected areas, so they are recognized by law. So they are established by law. But hotspots, these are not recognized or established by any law. So it is not formally recognized. So these are some of the hotspots of the world. South America, Africa, Southeast Asia, East Asia. So these are the 36 hotspots in the world. Then biodiversity hotspots in India. So these are the criteria. So to qualify as a hotspot, the area or the region it must qualify these two criteria. The region should have at least 1500 species of vascular species. That means it should have endemic species, and it must have lost more than 70% of its original habitat or the vegetation. Already we have studied this. So according to following these two criteria, so there are four hotspots in India. One is Himalayas, then Indo-Burma region, the Western Ghats, then Sunderland. So the Himalayas, that means hotspot region, it may not have only one country or nation. It may extend to more than one country. So the Himalayas, it contains East India, Northeast India, Bhutan, central and eastern part of Nepal. It has 163 endangered species, and it includes Asian water buffalo and one-horned rhinoceros. So here, 3,160 plant species are endemic. Then Indo-Burma, it covers India and also Burma. This is Indo-Burma. Then Western Ghats. So Western Ghats, that is present in India, in the western side of the Indian peninsula, and Odisha it is in eastern Ghat, and on the western Ghats, Tamil Nadu, Kerala, these states, and it is 325 number of endangered species. Then Sunderland, these hotspots, it is in Southeast Asia. So it covers Singapore, Thailand, Indonesia, Brunei, Malaysia, and also some parts of India in Andaman and Nicobar Islands. So in India, the Andaman and Nicobar Island, it is included in this hotspot. So here 15,000 endemic species are present. So there are many indices or index, plural of index is indices. So there are many indices, living plant indices, ecological footprint index, city development, human development, environmental sustainability index, like this. So in Singapore, you can see here, it is made an index and it is known as city index, because Singapore, it is a city state. The whole city consists of a country. So the index, it was devised by Singapore. It is city biodiversity index. So in city biodiversity index, so you can count the number of birds. So, but for biodiversity index, so it would be easier if biodiversity could be measured by quantity of birds in a forest. But biodiversity index, measurement of biodiversity index, it is not so easy just by calculating the number of birds. So because the ecosystems there very complex. So calculating biodiversity index, it is not easy. Why it is not easy? Because the ecosystems there very complex. So generally we believe that the biodiversity, it is equal to species diversity. Commonly, so there is a common belief that if there is species diversity, there is more number of species, then there is some biodiversity is more. But biodiversity is not a number of games. The quality is more important than the quantity. So the biodiversity, it is not about the only about the number of species, how many number of species, or the species richness or species evenness, but also the interaction between the species. So it is necessary to measure the abundance of all organisms over space and time using taxonomy. So one criteria is to measure the number of species, how many species are present by taxonomy. Also so that is morphological characters or taxonomical characters. But also we have to measure the functional traits. Functional trait means what type of organisms are present, what are their roles in the ecosystem, what are their interactions, and the functions and the interaction of the organisms, they are also very, very important. Just like the nitrogen fixing plants. How many nitrogen fixing plants are present? Because if more number of nitrogen fixing plants will be present, then the nitrogen content of the soil of the ecosystem, it will be more. Similarly, the interaction between the organisms, like the parasitism, how many parasitic organisms are present, and what is the competition, the competition for light, competition for water, competition for space, competition for minerals. Then how the organisms are dependent, or one organism is dependent on another organism for pollination. So these interactions, they are also very, very important when you study biodiversity. So in biodiversity, not only we study the number of species, so that is also important, but another important thing is the functional aspect, their relationship, what are their role in the ecosystem. So these are also very important. This species richness I have already explained. When you find the number of species richness, and species evenness or species abundance. So if in any area you find how many number of species are present, then this is known as species richness. And the individual A species, if there are 5 species, species A, B, C, D, E. So species A, so it is the species A, it is a number of individuals present in that area. So we have to calculate the how many number of individuals are present in that particular area. If 5 number of individuals are present, uh, 5 individuals of species A present in that area, then that is known as the species abundance of species A. So that is species abundance. There is also relative species abundance. Relative species abundance means what is the ratio of the abundance of a particular species to the total abundance. So if there are 5 species, for example, there are 5 species. Actually, in a ecosystem, there will be many number of species. But in example, if we take 5 species, A, B, C, D, E. Then species A, it has some individuals, for example, 5. If species B, it has 13. If species C, it has some number. If species D, it has some number. And if species E, it has some number. Species A has 5 number of individuals. So that is the abundance of that species. Hello?
କିନ୍ତୁ ମାମୁଁ ଯୋଉ କାଳିଆ ନଥିଲା ସାନ ବାପା ଆଜି ଆର୍ଥି ହେଇଗଲା ତ ଚାରିଟା ବେଳେ।
Anybody asking anything? So we can calculate the relative abundance. That means first we have to calculate the abundance of species A. Species A abundance is 5. If the total abundance of species A, B, C, D, E, and E, if it is 29, then the ratio, that means 29 divided by the abundance of species A 5, and that will give you the relative abundance. So it will be a fraction, and from that we can calculate the Shannon index. The Shannon index, it is minus of summation Ni divided by capital N into log again Ni divided by capital N. So Ni, that is the abundance of a particular species. For example, species A. Then N is the total number of species. So this is the Ni divided by N. You have to multiply with log of, again multiply with the log of Ni divided by N. Then it will give you a number. And this number, and this is the known as the Shannon index. So if the value of the Shannon index it is less, then it has less diversity, and if the value of this Shannon index it is more, then it has more diversity. Okay, the time is out. If anybody wants to ask anything. Okay, we shall end the meeting.